This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1152, recorded on September 26th, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from New York, Daniel Griffin.
Hello, everyone.
With a mystery bow tie, some kind of molecule.
It's actually, it's Creutzfeldt-Jakob disease again, right? With the prions on there.
Yep. The misfolded proteins, right?
Yes. It's actually, I don't know how new it is, but there's a test now where they actually take the misfolded proteins and then they put them with the native proteins, and almost like a PCR, it's like a chain reaction. All the other proteins misfold. Right. So,
yep. It's called shaking and quaking.
The FAST test. So,
yeah, it's really cool.
Yeah, for those of you studying for your infectious disease board, that's the right answer. So, all right, let's jump into it. We have a lot today. I'm going to start with an entertaining quotation from Benjamin Franklin. In wine, there's wisdom. In beer, there is freedom. In water, there is bacteria. And I have my my bacteria-laden water here. All right, so we'll start off with mpox. So let me jump in with this. So we have this article in a recent CIDRAP. I'm going to actually quote CIDRAP a couple times. Going to shine a little light on this publication out of University of Minnesota. But CIDRAP published September 19th, entitled, "Amid New Mpox Outbreak, Studies Suggest Waning Protection of Jynneos Vaccine." I'm sure Vincent will have some comments on the waning comment here. But here we read, Jynneos vaccine wanes significantly over the course of a year, raising new questions about just how protected vaccinated people are against reinfection and if booster doses of the vaccine are needed among at-risk populations. So the study, which we're going to discuss, was released alongside an alarming development in the globalization of mpox. More than 21,000 cases of virus have been recorded in the past year in the Democratic Republic of the Congo, UNICEF noted. On August 14th, the WHO declared the DRC outbreak a public health emergency of international concern. In part, I want to comment, about 60% of cases are in children 15 years and younger. So the majority of the cases are in children 15 years and under. Now, I know this led my my cousin, who I will mention later as well, to ask, well, wait a second, how can all these children be getting it? Now, in the DRC, children under age 15 account for about half of all the suspected cases and 80% of the deaths. So how does that happen? Because how is this spread, Vincent, right? Isn't this like a particular sexually transmitted disease of a certain population?
Well, it doesn't have to be. It can be close contact, which kids do with their parents, right?
Yeah, lots of close contact. We even think there's animals involved in the transmission. So, I just want to raise, the concern here, because this particular clade of mpox that we're talking about, it's, as we see, it's predominantly a disease of children. We're seeing more than 21,000 cases. We're seeing it spread to other countries. And things are not going well with mpox in Africa. We read in Reuters that the number of mpox cases in Africa has surged almost 200% compared to data a year ago. Deaths have increased 38.5% compared with the same period a year ago. As per the African CDC, we can say today that mpox is not under control in Africa. We still have this increase of cases that is worrying for all of us. John Kaseya, Director General of African CDC, told a weekly briefing. So I'll leave a link into that. But what's this study? Well, it's a preprint posted on medRxiv. Rapid decline of mpox antibody responses following MVA-BN vaccination. So that's the That's the modified Vaccinia Ancaria Bavarian Nordic vaccine. And this comes out of Dan Baruch's group up in Boston. So a little background here, but Dan Baruch is someone I think that we rely on. So I think it's just a matter of time before this is going to be a peer-reviewed publication. But here's where we are. So the orthopox virus genus consists of 12 viruses, including smallpox, mpox, vaccinia viruses. Now, the replication incompetent modified Vaccinia Ancaria Bavarian Nordic vaccine, this is JYNNEOS, was developed as part of the U.S. Strategic National Stockpile Program for deployment in event of smallpox outbreak. And the CDC has recommended this JYNNEOS vaccination to protect against mpox during the 2022 Clade 2B mpox outbreak. Now, due to limited supply, the recommended JYNNEOS administration was modified, right, from that 0.5 ml subcutaneous to this 0.1 ml by the intradermal route. So here, these investigators performed an observational study in 45 adults who received the JYNNEOS vaccine or had confirmed diagnosis of mpox infection at Beth Israel Deaconess Medical Center up in Boston. They assessed serum antibody and T-cell responses for 12 months. Right. So, what's the story here? We're looking at vaccinated. They're also going to compare it to people that had been infected. So we're going to get data on both. I'm going to, of course, focus on the mpox vaccination folks, but they're going to look at serum antibody, or they looked at serum antibody and T-cell responses. That'll make you happy, Vincent, for 12 months following either a 2-dose or a 1-dose vaccination delivered by either the subcutaneous or the intradermal route. They assessed median antibody ELISA titers to these different mpox, it's really monkeypox virus antigens, M1R, B6R, A35R, A29L, H3L antigens. And, I say monkeypox virus, right? Because we can rename the disease pretty quickly, which we did to mpox. But those viral taxonomists, we're still waiting on them. So it's still the mpox virus. So they looked at these ELISA titers, right? These antibody titers to these antigens, but they also looked at T cells. So they're going to measure interferon gamma CD4 positive and CD8 positive T cell responses. And they're going to do that with intracellular cytokine staining assays. Now, we're going to walk through the data a little bit here. Serum antibody titers following vaccination have been shown to correlate with protection against monkeypox virus challenge in non-human primates, whereas the CD4 positive and the CD8 positive T-cell responses did not correlate with protection. So sort of suggesting based on that, the potential relevance of measuring these serum antibody titers following vaccination in humans. So median binding antibody ELISA titers to mpox antigens peaked at week 3 following 2 doses of the JYNNEOS, but then declined at 12 months. And they've got data here for the 2-dose approach. They've got data here for the 1-dose approach, and really kind of similar. We get a peak, and then we get this what we've referred to as a contraction, an expected contraction of those antibody levels. With the 2-dose, you're going to end up with about a log higher or so median IgG titer, and you're going to see this contraction over time. Now here's the other thing I can say. So that's just total IgG. Now here's the other. I think this is important. Mpox serum neutralizing antibody titers. So these are the IgGs that are neutralizing, were detectable in only a few participants following 2-dose or 1-dose vaccination, and they actually show us the data for that. Okay, so we don't get the T cell data. They just tell us, right? I guess that's going to come when they get accepted. They tell us low peripheral interferon gamma, right? So that's our signature Th1, memory CD4 positive and CD8 positive T cell responses were detected to vaccinia infected target cells by intracellular cytokine staining at 9 months following 2-dose and 1-dose vaccination. They give us these median CD4 responses. They give us median CD8 responses, but we're not detected by these mpox peptide-specific ELISpot assays, and they don't show us that data. So they bring it all together. Vincent and I are going to discuss what all this means. They conclude, taken together, these data suggest that protective immunity may be waning in individuals who are vaccinated with the Jynneos vaccine in 2022, and that boosting may be required to maintain robust levels of protective immunity.
Okay, what's wrong with this?
You tell us, Vincent, what's going on?
Immunization or infection, antibody levels always decline. That's the way the immune system works. So here they're looking at baseline. 12 months later, it goes down. That's what you expect. If you did this with SARS-CoV-2, influenza, polio, you'd find the same thing.
Yep.
The fact that there are antibodies at 12 months means there are memory B cells present in these people. And what that means is when they get infected with monkeypox virus, they're going to make antibodies and the titer will go up. And that will protect them against mpox, the disease. So there is no reason. This is ignoring fundamental immunology. I'm really surprised that Dan, because he knows better, antibody levels always go down. We depend on a memory response to protect you. We don't depend on high antibody levels circulating all the time. The same thing happened with COVID vaccines, Daniel. After that first round of vaccinations, the titers were going down. Oh, they're failed. They're waning. They're no good. Oh my gosh, it's all over again here. So folks, don't worry about it. I think the key here is clinically, are these people protected? I don't care about the antibody titers, really. I want to know if they're clinically protected, and that's not done here. So
yeah, I think that's really, I mean, so one is the sky's not falling. This is basic immunology. You vaccinate, you get a peak in the antibodies, they contract over time. The question is, and it's sort of disease specific, like for instance, with meningitis, we need to keep those antibodies high, so we have to give periodic shots. A lot of other diseases, there's a certain amount of time for the memory cells to trigger, the elevation of those antibodies. And that's really what we're going to need to track is, okay, so it's a year out, and we have a fair, number of folks here in the United States that are a year out from last summer's vaccination. Are we going to start to see cases, or, do we just, does this become a once a year vaccination in certain populations? That's probably great for, Bavarian Nordic. If it's a once a year, it's a little bit more of a challenge, logistically. Who knows? Maybe, maybe the malaria vaccines that we've been discussing on other of our podcasts will need to be a yearly thing as well. So
I hope that upon peer review, the language is toned down, because to say that this may indicate that antibodies are that the vaccine is not working, that's not right. You can't make that conclusion.
Yeah, you sort of have to say, antibodies are contracting as expected, as we always see, and then sort of, we'll need to follow to see whether or not there's a clinical. Yeah. But I do like the language, could be even a little softer, I guess. Okay, a little more here on, and this, people may not realize this, right? So up until I'm about to say this, Bavarian Nordic's vaccine was really approved just for adults, but now we hear the Bavarian Nordic Jynneos vaccine was approved into adolescents, so 12 to 17 years of age. What about under 12? Well, Bavarian Nordic is preparing for a clinical trial to assess the immunogenicity and safety in children 2 to 12, right? I mean, that's what we need. I think we talked above that this is primarily affecting children. So we want to look at the immunogenicity and safety in children 2 to 12 years of age. This will probably be a bridging study, right? Where you make sure it's safe, you make sure you get the bridging, meaning you get these antibody responses. And this trial is partially funded by the Coalition for Epidemic Preparedness. So that's CEPI, Preparedness Innovations. And that's hopefully going to start in October. All right, it gets better or worse. According to media reports that cite government and health sources, India has reported an imported clade 1b mpox case, making it now the third country outside of Africa to report this clade. The patient is a 38-year-old man from the Malappuram district in India's Kerala state, who traveled from the United Arab Emirates, so Sweden, Thailand, and now India. And to close this section with maybe some more good news, I don't know, mixed here. On Tuesday, the 24th of September, we heard that Bavarian Nordic gets $63 million U.S. government order for the vaccine. And this is going to be a million freeze-dried vaccines from this Danish biotech company. More sort of support here. CIDRAP out of University of Minnesota had a news brief, they called it, and I get these emailed to me every day, where they report that at the United Nations General Assembly meeting, I watched a little bit of this, U.S. President Joe Biden announced new support for Africa's mpox response, including the 1 million vaccine doses and at least $500 million in further support. In the UN address, Biden called on other countries and groups to step up their help for Africa. And here I quote, "We call on governments, charities, and businesses to match our pledge and make this a $1 billion commitment to the people of Africa." The U.S. Department of Health and Human Services, the Administration for Strategic Preparedness and Response, is donating the 1 million doses to the international response. And it added that it is lending Bavarian Nordic, the maker of the Jynneos vaccine, 200,000 doses to ensure commercial supply to the U.S. market without diminishing the company's ability to fulfill these international orders and donations. African health officials have estimated the region needs at least 10 million vaccine doses for the current outbreak response, and a number of groups have also announced donations, including the European Commission, Japan's government, and actually Bavarian Nordic itself. So people are stepping up. Now, I was like, what can we do? So I was talking with, as I mentioned, my cousin Peter Dates, with whom I sail. I did donate to UNICEF, which I think we mentioned is the largest purchaser of vaccines and a major supporter of this effort. And I'm going to leave in a link here in case others want to do that. Now, since my cousin works in investment management, I asked him if it would be helpful to buy Bavarian Nordic stock, Vincent, because they're that Danish company making the Jynneos vaccine. And this seemed like something where I wasn't just giving away money, but maybe I was, responsible investing. So I also bought some Bavarian Nordic stock. My cousin seemed to think this might be helpful. He waffled there. But maybe we've got listeners who have better educated ideas. I'm certainly not trained in stock investing, and hoping this helps the company, but who knows?
I think it helps the stock market, right?
I don't know. Like maybe if the stock goes up, they'll be more like bullish on expanding. We'll get more vaccine. I don't know. I don't know what I'm talking about, but. All right, let's move into COVID. And, this is one of those times, Vincent, where we may have to talk about how we name the clinical update, right? Because people Google certain things, right? And we're going to talk about the fact here that SARS-CoV-2 was a zoonosis and spilled over into market. So we're going to talk about the article, Genetic Tracing of Market Wildlife and Viruses at the Epicenter of the COVID-19 Pandemic, published in Cell. And I sort of that lead in, right? Because, people like, when I Google Paxlovid rebound, I find articles about Paxlovid rebound. When I Google, the lab leak hypothesis, I find all this misinformation. So maybe people need to Google, I don't know, SARS-CoV-2 zoonoses. But so a couple highlights, and this really is a great article, I have to say, and hopefully Vincent and I can walk everyone through it. So they start off with some nice highlights. So highlight number 1 of 4, common ancestor of SARS-CoV-2 linked to Huanan market matches the global common ancestor. Number 2, wildlife mitochondrial DNA identified in samples from stalls positive for SARS-CoV-2. DNA from raccoon dogs, civets, and other wildlife species detected in market samples, and genotypes of potential hosts were reconstructed for retracing animal geographic origins. So then we get a nice summary. I mean, they really do a good job on this article. So start off with the summary. Zoonotic spillovers of viruses have occurred through the animal trade worldwide. The start of the COVID-19 pandemic was traced epidemiologically to the Hunan seafood wholesale market. So here they analyze environmental qPCR and sequencing data collected in the Hunan market in early 2020. They demonstrate that market-linked severe acute respiratory syndrome coronavirus 2, so SARS-CoV-2 genetic diversity is consistent with market emergence and find increased SARS-CoV-2 positivity near and within a wildlife stall. They identify wildlife DNA in all SARS-CoV-2 positive samples from this stall, including species such as the civets, bamboo rats, and raccoon dogs, previously identified as possible intermediate hosts. They also detect animal viruses that infect raccoon dogs, civets, and bamboo rats. And then combining metagenomic and phylogenetic approaches, they recover genotypes of market animals and compare them with those from farms and other markets. And this analysis provides a genetic basis for a short list of potential intermediate hosts of SARS-CoV-2 to prioritize for serological and viral sampling. So I have a really nice, clear graphical abstract, right, where they basically, they've got little maps, right, where you can see, the SARS virus, the market sort of viral hotspot, showing that not only are they actually identifying, where are all these viral samples, but even these, potential intermediate hosts. So they've got these cute little cartoons of the raccoon dog, the civet, the bamboo rat, and then a little bit about the DNA and the SARS-CoV-2 phylogeny. So this is really, I have to say, this is an important topic. So my plan is to actually spend some time today walking through this. I've got a few other articles that are sort of on hold for next week, so we could spend some time here. But let's go through, what we know so far and what this adds. So we'll start off. When cases of COVID-19 were identified, the question of where the virus came from and how it entered the human population started to be asked. Now, by the summer of 2022, the science was compelling that SARS-CoV-2 had entered the human population through multiple transmissions from wildlife at a market in Wuhan. Articles published during the summer of 2022 in Science, Volume 377, the Hunan seafood wholesale market in Wuhan was the early epicenter of the COVID-19 pandemic. Wildlife trade is likely the source of SARS-CoV-2, and the molecular epidemiology of multiple zoonotic origins of SARS-CoV-2 detailed the evidence around the timing and way that SARS-CoV-2 entered the human population. Within the Hunan wet market, the data statistically located the earliest human cases to one section where vendors of live wild animals congregated and where virus-positive environmental samples concentrated. It appears that infected animals were brought to the market, and this is where the jump to humans occurred. Based on sampling and analysis, the first zoonotic transmission likely involved lineage B virus around the 18th of November, 2019, and the separate introduction of lineage A likely occurred within weeks of this event. These findings indicated that it is unlikely that SARS-CoV-2 circulated widely in humans before November 2019 and defined the narrow window between when SARS-CoV-2 first jumped into humans and when the first cases of COVID-19 were reported. Spatial analysis within the market showed that SARS-CoV-2 positive environmental samples included cages, carts, and freezers, and these were associated with activities concentrated in the southwest corner of the market. This is the same section where the vendors were selling live mammals, including raccoon dogs, hog badgers, and red foxes, immediately before the COVID-19 pandemic. Multiple positive samples were taken from one stall known to have sold live mammals, and the water drain proximal to this stall, as well as other sewerages and a nearby wildlife stall on the southwest side of the market tested positive for SARS-CoV-2. There was an article, Surveillance of SARS-CoV-2 at the Hunan Seafood Market. I'll leave a link, published in Europe PMC in April 2023. And we have covered this extensively. TWiV has covered this issue on several podcasts, including the TWiV special, How the Pandemic Began in Nature, in 5 key points. I'm going to leave in a link. And there's actually been SARS-CoV-2 origin discussions on a number of previous TWiVs. So I'm going to leave in links to TWiV 1019, Eddie Holmes on SARS-CoV-2 origins, TWiV 1017, From Nature Not a Lab, TWiV 9095, Viral Origin Stories, that's TWiV 995. Also TWiV 940, Eddie Holmes again, Eddie Holmes in on viral origins, TWiV 876, Spillover Market with Michael Werribee, TWiV 762, SARS-CoV-2 origins with Robert Gary, TWiV 760, SARS-CoV-2 origins with Peter Daszak, Thea Colson, Fisher, Marian Koopsmans, TWiV 774, Christian Anderson, Robert Gary, and the deleted SARS-CoV-2 sequences. All right, so we've mentioned this before.
Yeah, we have. This is an important extension, though. They had new data, new analysis of. Remember that the sampling wasn't optimal. The sampling of the market was done, after it was closed and not really extensively. But this is what we have, and this is an extension of those samples and provides additional support for the notion, the hypothesis that the pandemic began here in the market, spill over from animals to people. It's quite clear.
Yeah. I mean, there's a lot of complexity here. So now that we've laid the groundwork, let's get ready to dive in. And Vincent, you can help me because this is a complex cell paper. So we're gonna start off with what do we learn from this paper? So number one, and this is, this is complex, but this is important. So SARS-CoV-2, so the virus, its genetic diversity linked to the Hunan market is actually consistent with market emergence. So here's what we read. So if the Hunan market was the site of origin of the transmission chains that led to COVID-19 pandemic, then the common ancestor of the market-associated viral genotypes should be equivalent to the common ancestor of the pandemic if you can get appropriate sampling. So to test this hypothesis, they assessed intra-sample variation of the SARS-CoV-2 environmental genomes from the Hunan market, assigned market sequences to virus lineages, and performed a phylogenetic inference to compare the genetic diversity of SARS-CoV-2 within the market to its genetic diversity globally. And they found that the most recent common ancestor, that's the MRCA, most recent common ancestor of market-associated genotypes, was actually equivalent to the MRCA of the larger pandemic. And they show us all this diversity. This is figure 1A goes through all this. Okay, so that's the first part. So they've got this most recent common ancestor analysis. So then they find increased SARS-CoV-2 positivity in and near a wildlife stall in the Hunan market, right? Sort of talked about some of this data before, but the Hunan market was sampled on multiple dates at the start of 2020 with different sampling trips having different purposes. They found that the rate of qPCR positivity was unevenly distributed within the market, with increased positivity in the, if you've been following, southwest sections. And one stall, Wildlife Stall A, stood out with a 30% PCR positive rate. So 3 of its 10 samples collected on January 12th, a cart, a hair feather removal device, and a sample collected from the ground were PCR positive for SARS-CoV-2.
I think this is important, that this is a big market.
Yeah.
And mammals were not sold everywhere. Mm-hmm. And they were sold in this part, the southwest corner, and that's where we see SARS-CoV-2 positivity. Yeah.
I mean, it's this nice southwest, nice map. They show us the hotspot. We get to see all the data here. So next, right? Next, mammalian wildlife species detected in 5 SARS-CoV-2 positive samples from a wildlife stall. So let's go through what this is. So environmental samples with viral RNA can also contain genetic evidence of the mammalian host that shed the virus, right? So we get a sample, we get the SARS-CoV virus, and then we can ask, hey, in the same sample, where did this come from? Maybe we can pick up an animal. So they developed and benchmarked a highly specific metagenomic pipeline for quantifying the abundance of animal mitochondrial DNA. That's the m little t DNA, mitochondrial DNA, in environmental samples. The 5 SARS-CoV-2 positive samples from wildlife stall A contained mitochondrial DNA from raccoon dogs, hoary bamboo rats, and dogs, so Canis lupus familiaris, and European rats. Amur hedgehog and Malayan porcupine mitochondrial DNA was present in 4 of the samples. Reeves muntjac and Himalayan marmot mitochondrial DNA was found in 3, and 1 sample contained masked palm civet mitochondrial DNA. And of these species, raccoon dogs, rabbits, and dogs are documented as susceptible to SARS-CoV-2, with raccoon dogs experimentally confirmed as capable of transmission and in vitro evidence of civet susceptibility.
So I think this is very important. Mitochondrial DNA can be used to identify what mammals were there, and they could, and then among them are mammals that can be infected with SARS-CoV-2.
Yeah, so they're really pulling this all together. And then this is kind of confirmatory. Like, okay, so we've got SARS-CoV-2, we've got the animals. Now, like, does this stuff all work? So what if we look for there should be other viruses that those mammals have, right? Because, and so really, I like this sort of a confirmatory analysis where they report that wildlife stalls and SARS-CoV-2 positive samples contained other mammalian viruses associated with the animal trade. And so the presence of animal viruses with predictable host ranges can provide evidence of animals productively infected with viruses in the Hunan market. So by mapping sequencing reads to a custom database of human and animal viruses with stringent filtering, they identified several mammalian viruses present in the market. So not only did they find the DNA of the viruses of the animals, so not only did they find the DNA of the animals, but they find the typical viruses that infect those animals. So they're identifying things like the raccoon dog amdovirus, the civet kobu virus, canine parvovirus, and the bamboo rat coronavirus, among others.
Yeah, I think that's a nice addition that these animals have other viruses. So can we detect them? Yeah, we can.
Yeah. So you find SARS-CoV-2, you find all these sort of lineage, that sort of fits into this really nice common ancestor analysis, really showing that you had quite a bit of diversity, right? So I'm trying to, the first sort of part of this thing, like, oh, what's the virus doing there so close to this lab? Well, it's not the virus. There's this incredible diversity, actually, genetic diversity in this market, really consistent with a bunch of animals were brought there that were infected. And, what do you sort of, the lab people, they didn't just have to make supposedly a virus, they had to make all these different viruses with this great tropism for all these animals. And then, so the first part sort of falls apart. The second is you've got this really nice, hotspot. You've got the right animals in that hotspot. You confirm that with the fact that you're also able to detect other viruses that affect those animals. So, why do we spend so much time on this? If you look through the list of times, it's important because, as we discussed, there's a lot of damage from this misinformation campaign. It's a misinformation anti-science campaign. And in TWiV 1140, we actually discussed that in the clinical update.
Yeah, the evidence in supporting a natural origin of SARS-CoV-2 continues to accumulate, and there's no support for any other origin. So individuals who promulgate other origins are not really being scientifically rigorous. So and it damages science, as Daniel says, because you generate mistrust for scientists in China, for example, which isn't justified in this case at all.
Yeah. I mean, here's the information. It's out there. We keep learning more. I mean, okay, so back in 2020, it was a reasonable question to say, oh my gosh, there's a lab nearby. It was this, a bat got brought there. Somehow there was a connection. It's overwhelming at this time. This is what happened. There's a lot of information here. All right. Moving into what is going on with COVID right now. It's now North Carolina's the hot spot. 4 to 5.9% of the total deaths in North Carolina are due to COVID.
Oh, so Daniel, that must mean that it came from a lab in North Carolina, right?
Well, my little brother lives in North Carolina and he's nothing but trouble. So I'm going to call Ben up because, maybe he's responsible. He has these big parties, you see, Vincent.
I see.
I'm only joking, Ben. My brother had nothing to do with this. Anyway. All right. So what about the wastewater? It looks like things, a little bit of a blip there out in the west, but most things look like they're moving down with wastewater across the country. I'm predicting, Vincent, a lull coming up in early November for about 7 days before it shoots right back up in December and January. But Vincent's predicting we're all good till next summer, right?
I think so. That's my prediction. I could be wrong, but I want to inject some optimism into this story.
All right. And the variants, right? It's all KP.3.1.1, but this is all JN1 lineage, right? So that'll be important when we talk about how well things go with the booster doses, right? And a lot of people are jumping in for those booster vaccines. We seem to be like team Novavax in my family. With my parents both got it on Monday, and my wife got it. A couple of my daughters have gotten it. I don't know if my son in England can get it. So if anyone's over there in England, let me know if there's access. But nice thing is, nobody had any noted reactogenicity, which was really nice. So everyone's getting their updated vaccines. All right. We will talk about, unfortunately, folks do keep testing positive. Early viral phase, we still have our guidelines. Number one is Paxlovid, recommended by the ID Society of America and the NIH COVID-19 treatment guidelines. I actually had a patient, I was consulting on a patient in the hospital today, and she was telling me her story. It was about 9 days ago. That she was diagnosed at an urgent care with acute COVID. They suggested she go get Paxlovid. She went. Apparently, the pharmacy told her that it would be $1,100 for her to get that Paxlovid. So she wasn't really sure what to do. She ended up not getting the Paxlovid. She called her pulmonologist, who, as she explained, does not believe in treating Paxlovid with those medicines. So instead, he increased her steroids, and now I'm taking care of this hypoxic woman in the hospital. I informed her that I take care of a lot of that particular provider's hypoxic patients during week 2 in the hospital, and she got the point. Okay, number 2, remdesivir. 3, molnupiravir. 4, convalescent plasma. And isolation guidance. I looked ahead. I saw we have some questions, so we're going to talk a little bit about isolation guidance in our Q&A section. Okay. Number 2, early inflammatory week. This is week 2. This is, for a lot of folks, that bad week when they might feel even more rotten than the first, when they get that hypoxemia, that cytokine storm, the early inflammatory phase, steroids at the right time in the right patient at the right dose. Number 2, anticoagulation. Number 3, pulmonary. And 4, remdesivir. This is updated data, right? So I know sometimes we kind of memorize like A, if A, then B, but we learn more and then A might become C. So the article, lower mortality risk associated with remdesivir plus dexamethasone versus dexamethasone alone for the treatment of patients hospitalized for COVID-19, published in CID. So the reason this is relevant is for a while, we sort of had this dogma of, okay, if it's past day 10, if they're in the ICU, if they're requiring a high amount of oxygen or ventilated, okay, maybe you missed your window for remdesivir. So here is this issue about, hey, maybe if it's that second week and they're on dexamethasone, let's see, adding the remdesivir, is that helpful or not? So large multi-center U.S. hospital database was used to identify hospitalized adult patients with a primary diagnosis of COVID-19. Also, they flagged it as present on admission, treated with remdesivir and dexamethasone or dexamethasone alone from December 2021 to April 2023. Patients were matched 1-to-1 using propensity score matching, stratified by baseline oxygen requirements. They do this Cox proportional hazards model to assess time to 14 and 28-day in-hospital all-cause mortality. A total of 33,037 patients were matched. Most of the patients were 65 years of age or older, so 72%. 78% were white and non-Hispanic. Remdesivir plus dexamethasone was associated with lower mortality risk versus dexamethasone alone across all baseline oxygen requirements at 14 days. So looking at no oxygen, we saw an adjusted hazard ratio of 0.79, so 21% reduction. Low flow, 0.70, so 30% reduction. High flow oxygen or non-invasive ventilation, 0.69, so 31% reduction. Invasive mechanical ventilation, ECMO, also 0.78, so 22% reduction. All this is with confidence intervals, favoring this. Similar results at 28 days. So really
it's
sort of
consistent with other studies showing that you can give remdesivir in hospital. It's not too late, right?
Yeah, so we certainly do it in the hospital. And the little twist here was we often said, if it's after day 10, if they're already in the ICU, and this was like sort of just straight remdesivir studies, you seem like you'd sort of missed your window. But it looks like if you've got them in the ICU, you've got them on steroids, it's still worth doing the remdesivir. All right. And just a little bit here, a little disturbing bit here on the late phase past long COVID, because we've got a really nice, it's actually another Cell paper on long COVID that we'll be talking about next week, among some other things. But I don't know if you saw this one, Vincent, but this one really bothered me. So this is the article, Changes in Memory and Cognition During the SARS-CoV-2 Human Challenge Study, recently published in eClinical Medicine. So I don't know if our listeners remember that human challenge trial. We had some trepidation about giving the SARS-CoV-2 virus to healthy volunteers. We didn't really fully understand the repercussions. So here, 34 young, healthy, seronegative volunteers were inoculated with wild-type SARS-CoV-2 under this prospectively controlled conditions. Volunteers completed daily physiological measurements and computerized cognitive tasks during quarantine and follow up at 30, 90, 180, 270, and 360 days. They're out for a year. Linear modeling examined differences between infected and inoculated but uninfected individuals. The main cognitive endpoint was the baseline corrected global cognitive composite score across the battery of tasks administered to the volunteers. Exploratory cognitive endpoints included baseline corrected scores from individual tasks. So, all right, let's go through the data. 18 volunteers developed infection by qPCR criteria of sustained viral load, 1 without symptoms, and the remainder with mild illness. Infected volunteers showed statistically lower baseline corrected global composite cognitive scores than uninfected volunteers, both acutely and during follow-up. And they give us the mean difference overall at different time points with significant main effect of group and repeated measures. sensitivity analysis replicated this cross-group difference after controlling for community upper respiratory tract infection, task learning, remdesivir treatment, baseline reference, and model structure. Memory and executive function tasks showed the largest between group differences. So what happened here? So in this study, the volunteers completed 11 computerized cognitive tasks in a fixed order on an iPad during 2 consecutive days pre-inoculation. So we get a baseline, each quarantine day, and each follow-up visit. And the 11 tasks included were motor control, object memory, simple reaction time, choice reaction time, 2D manipulations, this 4 towers, which looks at 3D visuospatial information, spatial span, which working memory capacity, target detection, which looks at attention and distractibility, a Tower of London, which measures spatial planning, they do verbal analogies, which measures semantic reasoning, and object memory, which measures the medium-term precision recognition memory. And you can actually look at this figure where you see that everyone's sort of, basically at the same baseline, and then you follow them out over time, and you see that the infected have this really significantly lower cognitive score all the way out to a year.
I don't understand why the score goes up for the uninfected, right, from baseline. Why would that be?
I think because you would expect when you repeat a test, people start to do better, right? Because they've done it before. So in general, and it's really nice that they have a baseline here because you'd say, oh, they're just about the same. Well, you're doing this task over and over. You should be learning. You should be getting better.
I see.
And you could see like the uninfected people, they actually get better. Like they come up to this really nice and they hold pretty much there. After a while, you could see like by the end of the year, they're getting a little kind of tired of doing this. But the infected people, they're like, they really get this. They don't really tend to like learn over time. So they found that the decrements in memory precision and executive function were the main contributors to reduced task scores following infection. So they found that volunteers who exhibited sustained viral load after inoculation with SARS-CoV-2 performed worse on a measure of global cognition than volunteers who did not exhibit the sustained viral load. And this deficit persisted up to a year after inoculation. None of the volunteers reported subjective cognitive deficits, so they didn't even realize that they had all these deficits.
So I never thought this challenge study was a good idea because of this. How do you know this wasn't going to happen? You didn't. This is an unknown virus. So this shows that it's not ethical. It was not ethical to do this. What do you think, Daniel?
Yeah, I mean, I share the same, concern. So here they took these young, healthy volunteers, and these are like, these are good folk. These are folk like, altruistic. I want to make a difference. What can I do? And now they're cognitively impaired. They don't even realize it, but when you test them, like this damaged their brains. Bad news. All right. No one is safe until everyone is safe, even these folks. I hope everyone will pause the recording right here. Go to parasiteswithoutborders.com and click donate. Every bit helps. That's what allows us to do this work. We don't accept money from big pharma and all these other sponsors. It's all you. It's all our listeners. We're now doing our Floating Doctors fundraiser. Actually, Ben LeBrat, the head of Floating Doctors, is going to be a guest on TWiV coming up soon. During the months of August, September, and October, we'll double your donations up to a potential maximum donation of $20,000.
It's time for your questions for Daniel. You can send yours to daniel@microbe.tv. Ginny writes, I received the excellent newsletters from Caitlin Hetelina, your local epidemiologist. I don't know, Ginny, maybe you shouldn't. In the most recent one, she answered a question like this. If my COVID antigen at-home test is positive for a long time, 14, 21, 28 days, am I still contagious? And here is the epidemiologist's response. Unfortunately, we don't have good data on this. But most likely you're still contagious. Wait a minute. She said they don't have good data, but she's making a conclusion? Okay.
Yeah.
COVID-19 antigen tests are very good at detecting infectious COVID-19 virus. Really? Really? PCRs, conversely, are good at telling you whether you have the virus regardless of infectiousness. Oh my gosh, she's digging it deeper and deeper.
Yes.
Be sure to wear a mask. And so back to Ginny, to her credit, she writes, is that right? And since antigen tests detect protein, are they very good at detecting infectious virus? I thought you needed something like a plaque assay. See, I've been paying attention.
Yes.
Or is it that you won't get the protein unless the virus is active? I thought the antigen test might see remnants even after the virus is no longer active.
Okay, so JD, thanks for writing us. This is a great chance for us to go through this, right? So we do have good data. I mean, it's actually amazing. The data we have for COVID-19, for SARS-CoV-2, is actually better than the data we have on almost any other pathogen out there. I mean, it's amazing the amount of money and resources and studies that have gone into this. So this is what we know. Unless you have significant immunocompromise, you get infected, you test positive, your symptoms start, that's day 0. After, we'll say, the first 5 days, 85% of the transmission events happen in those first 5 days. We still see about 15% of the transmission in days 6 through 10. Really, it's day 6, 7, maybe 8. We really don't even see much on day 9, 10. But after day 10, we really do not see transmission, period. It does not correlate with symptoms. Folks that go on to develop, that inflammatory phase, where they feel crummy, they do not become contagious again. We have studied this in, billion people, right? So we do actually have really good data on when you transmit to others. The tests are great for making the diagnosis, but they're not great for tracking whether or not you're contagious, right? So I still see to this day, it's day, let's say, 11, 12, 14, 21. People are testing to, quote unquote, be safe. If it's after day 10, you don't transmit to others. Those tests have no role in helping you make any kind of further decision along those lines. And yes, Jenny, you are correct. The antigen tests pick up the protein, they pick up the antigen, and they can remain positive for a very long period. The PCR also can remain positive for a very long period. And it doesn't tell you about the virus. It tells you about the RNA. It detects RNA. If you want to actually pick up, is there replication competent virus? You need a plaque assay.
And when you need virological advice, you need to talk to a virologist, not an epidemiologist. Yeah. Sorry. Mike writes, wrestlers and martial artists sometimes need to worry about things like ringworm and herpes gladiatorum. Do you think eventually we'd need to worry about mpox as well?
I'm not sure it's eventually, right? Wrestlers, there's a lot of skin to skin. There's a lot of close contact. Folks that have mpox are able to transmit from the time they start to feel sick, even before you can actually see any skin lesions. They continue to transmit until they have full intact skin in those areas. So no, I think that, we need to keep track because there may be some at-risk individuals participating in these activities, in the wrestling, and potentially it could get introduced into a population that way.
Alec writes, greetings from Cambridge, Massachusetts. Hearing the letter about the mRNA vaccine disinformation in last week's clinical update motivated me to write you. My partner recently got COVID for the first time. She's 67 and healthy. We both had gotten Paxlovid prescriptions last winter with difficulty in advance of traveling, so she was able to start Paxlovid after her first positive test. After a couple of days, she's feeling much better. Our neighbors warned her about Paxlovid rebound. Being a faithful listener of the TWiV clinical update, I said confidently that Paxlovid rebound is not a thing. She Googled Paxlovid rebound, and the first result was a blog post for laypeople from a medical school in our area that you may have heard of, titled Paxlovid Rebound, What You Need to Know, May 9th. It defines Paxlovid rebound, has sections, how to know if you're experiencing rebound and managing rebound. In short, it treats Paxlovid rebound as real. Buried in the 9th paragraph, it says, For people at high risk of getting very sick or dying from COVID, the benefits of Paxlovid far outweigh any risk of rebound. It mentions the large clinical trials you often cite that show no evidence for Paxlovid rebound. The only evidence of rebound is a small observational study from Harvard Medical School. The first sentence of the results section of this paper is, compared with untreated persons, N equals 55, those taking NR, N equals 72, were older, received more COVID vaccinations, and more commonly had immunosuppression. I'm shocked that one of the most famous medical schools in the world would publish an article that might discourage people from taking a potentially life-saving treatment based on a concern that probably is not real. What is to be done?
Yeah, this is tough, and I worry about a lot of times. I even hear these quote unquote thought leaders, senior people, and you could tell that they're actually not reading the science. They're actually quoting from the New York Times. I'm like, seriously, if you're going to be the chief of a division, if you're going to be the one putting out this advice, you should actually have the integrity to read the science and not just recycle what, sort of secondhand information from the media. We're not here to be an echo chamber of the media. We're here to actually take the time and give reliable, credible advice.
As Mark Crislip always said, read the literature.
Yes.
Carmen writes, my son, now a 28-year-old New Yorker, was exposed to TB in South Africa at 7 years old when his father took him for a prison visit, as well as staying in the townships. He had a big new positive PPD. 4 months later, he could not take INH because of bizarre hyper behavior changes. He's been healthy since. If that TB exposure were to become a health issue for him, what ID team in New York City would do appropriate tracking of TB resistance to factor into treatment for him? Been thinking to ask for a while.
Yeah, so I think there's two aspects. So one is that, if there was this issue with INH, there are other, rifampin would be another potential way of treating that latent TB, right? Because, 5% lifetime risk of a relapse, so 1 in 20. Columbia has a great department that would be happy to help.
And Connie writes, thanks again for your podcast. I recently read Hedalina's newsletter that it might be beneficial for people to get a pertussis vaccine every 10 years. She wrote and referenced an article from the UK that the pertussis vaccine that's been used for decades loses its effectiveness over time more so than expected. Can you give me any information? I'm 75, got a Tdap booster shortly before my oldest grandchild was born in 2016. Do I need another one in the near future?
Wow, I guess everyone's reading Hedalina's newsletter. We we do actually recommend the Tdap every 10 years. I'm not sure if I would say, loses its effectiveness more than expected. But and what we also recommend is it's really the pertussis booster every pregnancy. Yep. So this is something you want to do more than once in your life.
That's TWiV Weekly Clinical Update with Dr. Daniel Griffin. Thank you, Daniel.
Thank you. And everyone, be safe.